Hello, welcome to Module 1, Lesson 2 in BIO100A related to cell division. This is a refresher on the types of cell division for prokaryotic and eukaryotic cell, as well as the cell cycle phases. For more information, please refer to Chapter 8 of the BIO100 book. First of all, why do cells divide? Well, those organisms that are unicellular, which means one cell is the organisms, they divide for reproduction, and this entails both prokaryotic organisms, which are all unicellular, and some eukaryotes. In the cells of multicelled organisms, and that includes us, for example, the cells may divide for growth, reproduction, but also for repair. In the next slides, we are going to revisit binary fission, which is the way how prokaryotic cells divide. And we are also going to look at mitosis, which is the cell division of eukaryotic cells for growth and repair. We are not going to cover this time meiosis, which is a special kind of cell division to produce reproductive cells. And we are also going to look at the cell cycle phases.
Most prokaryotes, the bacteria and archaea, have less DNA than eukaryotes. They carry their genetic information in a singular circular chromosome, a closed loop of double-stranded DNA that's attached at one side to the cell membrane. And when it's time for them to reproduce, they use a method called binary fission, which means division in two. This process begins with replication, the method by which a cell creates an exact duplicate of each chromosome. The two newly created circular chromosomes attach to the inside of the plasma membrane, each at a different spot. The original cell, called the parent cell, then pinches in somewhere between these two spots until it divides into two new cells called daughter cells. Each of the daughter cells has an identical two-stranded copy of the original two-stranded circular chromosome. Binary fission is considered asexual reproduction because the daughter cells inherit their DNA from a single parent cell and thus are genetically identical to the parent. The figure shows the phases of the cell cycle. The two main phases are interphase, during which the cell grows and prepares to divide, and the mitotic phase, or M phase, during which division occurs. Interphase is further divided into three distinct subphases, described on subsequent slides. Gap 1, G1. During this period, a cell may grow and develop as well as form its various cellular functions, making proteins, getting rid of waste, and so on. Most cells inhabit the Gap 1 phase the majority of the time. However, some cells enter a state called G0, which is a resting phase outside the cell cycle in which no cell division occurs. Gap 2, G2. Significant growth occurs in this phase along with high rates of protein synthesis in preparation for division. Mitotic phase. This period begins with mitosis, a process in which the parent cell's nucleus, including its chromosomes, divides. Mitosis is generally followed by cytokinesis, which may begin before the end of mitosis, during which the cytoplasm is divided into two daughter cells. Mitosis has just one purpose: to enable existing cells to generate new genetically identical cells. There are two different reasons for this need. 1. Growth. During development, organisms get bigger. Growth happens in part through the creation of new cells. A plant root, for example, may grow half an inch per day. The root is one of the fastest growing parts of a plant. 2. Replacement. Cells must be replaced when they die. The wear and tear that come from living can physically damage cells. The daily act of shaving, for example, damages thousands of cells on a man's face. Fortunately, the layers under assault during shaving are made up primarily of dead cells. These dead cells help protect us from infection and reduce the rate at which the underlying living cells dry out. The cells living beneath the layers of dead cells are produced at a high rate by mitosis. Mitosis occurs in just 4 steps. It cannot begin, however, until after an important event occurs during the previous portion of the cell cycle, interphase. During the synthesis portion of interphase, all of the chromosomes replicate. Mitosis then begins with 1. The condensing of the chromosomes, which during interphase are all stretched out and stringy. 2. Next, all of the duplicated and condensed pairs of chromosomes move to the center of the cell. 3. Each chromosome is pulled apart from its duplicate. 4. And finally, new cell membranes form around each complete set of chromosomes, and the cytoplasm duplicates as well. Where once there was one cell, now there are two. Throughout the cell cycle, except for mitosis, chromosomes are uncoiled and spread out in a diffuse way, like a mass of spaghetti. And because they're so stretched out, they're not dense enough to be visible under a light microscope. However, just before mitosis begins, two important events occur. 1. The chromosomes replicate, becoming two identical linear DNA molecules. The two DNA molecules are held together at a region on each molecule called the centromere. Throughout mitosis, until the centromeres separate, each of the identical DNA molecules is called a chromatid. Together, the two are called sister chromatids. 2. The sister chromatids begin the process of condensation, in which they coil tightly and become compact, in contrast to the uncondensed and tangled state of the chromosomes prior to replication, during most of interphase. Because the sister chromatids need space to separate, the membrane around the nucleus is dismantled and disappears early in mitosis. At the same time, a structure called the spindle is assembled. The spindle is made mostly of hollow tubes of protein called microtubules, which are part of the cell's cytoskeleton. They resemble a group of threads stretching across the cell between its two ends, or poles. In animal cells, the threads originate and spread out at each pole from structures called centrosomes, which contain a pair of centrioles and a mass of proteins that anchor the microtubules. These threads, known as spindle fibers, attach to the centromeres and pull the sister chromatids to the middle of the cell. During mitosis, they'll eventually pull the chromatids apart as cell division proceeds. The actual process of cell division occurs in 4 stages. 1. Prophase. Following replication, the sister chromatids condense. Prophase begins when the sister chromatids have condensed sufficiently to be seen with a light microscope. At this point, the spindle forms and the nuclear envelope breaks down. 2. Metaphase. The chromatids congregate at the cell center. After condensing, the pairs of sister chromatids line up at the cell's center, pulled by the spindle fibers attached to a disc-like group of proteins called a kinetochore that develops on the centromere of each pair. At the end of metaphase, all the chromatid pairs are lined up in orderly fashion, straddling the center in a single-file congregation that is called the metaphase plate. The chromatids are at their most condensed during this part of mitosis. The third phase is anaphase. The chromatids separate and move in opposite directions. In anaphase, the spindle microtubules attached to the centromeres begin pulling each chromatid and the sister chromatid pairs toward opposite poles of the cell. From each pair of sister chromatids, the centromere splits as one DNA molecule is pulled in one direction and the other identical DNA molecule is pulled in the opposite direction. At the end of anaphase, one full set of chromosomes is at one end of the cell, and another identical set eventually occupy the nucleus of each of these two new daughter cells that result from the cell division. 4. Telophase. New nuclear membranes form around the two complete chromosome sets. With two full identical sets of chromosomes collected at either end of the cell, the parent cell is prepared to divide into two genetically identical cells. In this last stage, called telophase, the chromosomes begin to uncoil and fade from view. Nuclear membranes reassemble, and the cell begins to divide in two. The process of mitosis is generally accompanied by cytokinesis. During cytokinesis, the cell's cytoplasm is divided into approximately equal parts and the cell divides, with some of the organelles going into each of the two new cells.
So what did you learn today? In summary, prokaryotes, such as bacteria, divide by a process called binary fission. Eukaryotes, actively dividing eukaryotic cells, have a cell cycle which consists of several phases, one of them being mitosis, which is the type of cell division that is done for growth and repair. And finally, mitosis consists of several distinct phases. Thank you.